I'd just like to begin with a land acknowledgement. My research takes place on the campus out at UBC. That's the territory of the Musqueam people. But today I'm coming to you from the Okanagan, and this is an unceded territory of the Syilx Okanagan people. And I'm thrilled to be able to be delivering this presentation to you all. It's really an honor. I have no conflicts of interest other than that my lab studies gene therapy and genome editing that I'm going to be talking to you about today. And so we receive funding from provincial, federal, and international granting agencies. So I put this slide in just to remind you and I that I'm going to be talking about LNP mediated in vivo delivery of genome editors. But I'm going to really start it out with a story of why. And, so I'm going to build it up. So I will get to this, but I'm going to start with a little bit of background. So why gene therapy and why genome editing for genetic diseases? So I grew up in Penticton. I was born here in the Penticton Hospital. And I think my interest in genetic diseases started literally on the day I was born. The little girl born in the room next to me was a family friend, or is a family friend. And she was born with a really serious genetic disease. And we both sort of grew up leading very different lives. She was always interested in where she would have been in school, but was not allowed to or not able to attend the regular school. And so I've always been interested in genetic diseases, and I've come across a few patients in my life in elementary school, and then as I grew older. And just so and I'm sure a lot of us here on this call have also had personal interactions with people with genetic diseases, and maybe that's why a lot of us are so interested in trying to treat these diseases, because from a big picture, even a small benefit for some of these diseases can have a huge impact on the patient and on their families. And there's an estimated now up to 10,000 different rare genetic diseases. The number is pretty astounding in how many people it affects. So some estimates are around about 1 in 40 people, and the cause of 20 to 30% of infant deaths, and accounting for half the cost of pediatric practice. But unfortunately, and I'll go over this a little bit later too, but there's very few therapeutic options for most of these genetic diseases. And for the few that are emerging, they're extremely expensive. So I'm speaking to you guys today from Summerland, and this is actually exactly the place where I first learned how to do DNA sequencing. It's a beautiful campus, the Summerland Research Station. It's over 100 years old. This building is, of course, newer than that. But I learned how to do DNA sequencing there, the old-fashioned way, and we were sequencing cherry tree viruses, if you can believe it. And it took me about 2 weeks to sequence 300 base pairs of sequence. I wasn't very good, but this was actually that's pretty normal. And as I said, I was interested in genetics. I went on to do a master's degree after an undergrad out at UBC. And it literally took me 2 years to narrow down the region of one disease-causing gene from about 5 million base pairs to a 1 million base pair region. And it was basically part of a 20-year project trying to identify the disease-causing mutation for a rare genetic disease. And so you can expect that I was really keen on new advances in DNA sequencing as they emerged, because I calculated it would take me about 50,000 years to sequence a single genome at the rate I was doing it at the Summerland Research Station. And then in 2001, the first draft of the human sequence came out, and just this year, back in March, the final, the most up-to-date sequence of the genome was announced that filled in all the gaps. And today we can sequence an entire genome in a single day, and the cost is probably less than $1,000 now. And I put this slide in for the non-genetics people just to maybe hopefully capture my interest in gene therapy and especially genome editing. And so the genome, a human genome is big, it's huge, but it's hard to kind of conceptualize it. And so here I've drawn it as if I had typed it out in 10-point font, something on a regular piece of paper. And it would stretch all the way across Canada to Thunder Bay, and then the second copy in your cells, one from your mother, one from your father, the second copy would stretch from Thunder Bay all the way out to Newfoundland. And important point is that a single nucleotide difference in that stretch of DNA can be the difference between, a relatively normal life or being born with a genetic disease and spending the rest of your life trying to work around that genetic disease, or maybe not surviving. As I said, I've been always interested in DNA sequencing, and our lab installed the very first next-gen DNA sequencer at BC Children's Hospital. And I helped set up the very first pilot clinical genome sequencing at BC Children's Hospital. This was called TIDE BC. And then it later transitioned into the Causes Clinic. And these were with Clara Van Carnebeck and Jen Friedman. And they were so interesting to be a part of because, instead of that 20-year odyssey of looking for a disease-causing gene, we could do it in a matter of weeks or even days. So it was wonderful. We discovered many new disease-causing genes. In one paper, we quantified how many diagnoses we came up with, and we found that we had diagnosed 68% of patients that were otherwise had no diagnosis. Everything else had been tried. And this was really not specific, of course, to BC Children's Hospital, but people have been doing this all around the world. Now there's all kinds of clinical genome sequencing programs, and all, I think, being very successful. So it's great. It's, if you're interested in trying to identify the cause of genetic diseases, we've really come a long way. And now there's even newer DNA sequencers coming on the market that are even faster and more accurate because they can read much longer lengths of DNA. So that's been great. But on the downside, even today, most genetic diseases have no treatment. I'm not saying that the clinicians aren't trying their hardest to come up with ways to treat symptoms or to come up with really innovative strategies to treat these genetic diseases. They really are, and it's really amazing to see these clinicians and what they can do. But there's very few clinically approved treatments for most genetic diseases. And so for me, why gene therapy for genetic diseases? I've met many people like these people listed here. They all have different genetic diseases, all with completely different symptoms, different onsets, different severities. All of them have families that are keen to help support these patients developing new treatments. And Jeff here on the far right is a colleague, and we did our postdocs together. And he's working on treatments for Huntington's disease. And to me, with a background in genetics, what I've always been fascinated with is that you have all these completely different diseases, but they really all have the same root cause of different mutation in the DNA. And so if we could treat those, or somehow treat those, we could potentially treat thousands of different diseases using a very similar way. And that's really the underlying rationale for gene therapy, and as I'll come to later, genome editing. And at a high level, there's different strategies of doing this, and one sort of simple way is basically just flooding the cell with new copies of the disease-causing gene, and that's called gene augmentation. Another way is at my PhD defense this morning, a student was inhibiting a dysfunctional gene using siRNA. Another one that we'll talk about is genome editing, which I like particularly because it's just so precise, fixing that exact mutation that's causing the disease. And it's exciting too because it was almost exactly 50 years ago this year that sort of gene therapy was really first proposed in a formal publication by Theodore Friedman and Richard Froblin. So going back to gene therapy, and I want to talk to you about my history of gene therapy. I came across patients that suffered from this disease called type 1 hyperlipidemia, or LPL deficiency, named after the gene that has the mutation causing the disease. And I teamed up as a postdoc with Michael Hayden, who he was well known for discovering the disease-causing gene for this disease, the lipoprotein lipase gene. And I was interested to work on this project because he was interested in using a viral vector method that was quite innovative and new at the time. And so I wanted to tell you about that story. So this is the clinical characteristics of the disease. Basically, your body is not able to take fat triglycerides from your blood into your cells. And so this is normally occurs in your muscle and your adipose tissue and your heart. And so as a consequence, this fat can basically leak out of your skin and causing these xanthomas that are painful. You can get pancreatic necrosis, which can be lethal. And if you take a blood sample, this is what the plasma looks like. These patients, their plasma looks like milk. And so this is just showing, a little bit more detailed about it, but essentially it's just that you can't break down these fats in your blood. And it's a very Canadian disease. It has the highest worldwide frequency in Eastern Quebec compared to the rest of the world. And this was just because of a founder effect from some of the settlers of Quebec. And I blame some of my relatives who are from Scotland and from France. And these people would come to Canada and have huge families. And my ancestors, I know that my great-great-grandmother was the oldest of 19. And that was pretty normal. And so there's a small number of founder mutations that are causing this high incidence in Eastern Canada. And so when I started in the lab to work on lipoprotein lipase gene therapy, we had already established that lipid-lowering drugs really had no effect on these patients. People had tried enzyme replacement, which is just purifying the LPL and then injecting it, and it literally did nothing, just because the half-life of the enzyme is so short. And so the standard treatment for patients has been basically eliminating dietary fat, because if you don't input any fat, then there's no fat to build up. But this is tricky, and even there's patients at Children's Hospital today that are really trying their hardest to manage the symptoms, and are just unable to do it as well as the clinicians would hope. They're really trying their hardest. And so we were interested in developing a gene therapy for this because of the rationale that I had mentioned before. And it was a relatively new approach of using a viral vector to deliver the gene. So this would be a gene augmentation approach. And the reason for this choice of a vector was that this is a single-stranded DNA virus called adeno-associated virus, and it's I think it is the smallest known virus that will infect human cells, and it's considered non-pathogenic. And it's actually such a weak virus that it can't even replicate by itself. It requires other co-infection of other viruses. So it's essentially tricking this virus by switching out the DNA of the virus with the gene that we want to deliver. And what's especially good about this AAV approach is that the genomes of the virus would get into the cell and essentially just stay there for the life of the cell. So it was very long term. Whereas at the time, back in 2000, sort of the state of the art gene delivery systems were using adenovirus, which would last only a few days. And they had quite high toxicity compared to AAV. And so in choosing the serotype, I, of course, just looked in the literature and saw, okay, well, you can see from this figure, this is an in vitro figure of how well these different serotypes work. There was only a few, 6 or so serotypes that were known. So, of course, I picked the AAV2, which was the best one by far, and we used that. And so I'm showing here results of just us trying to deliver GFP, this green fluorescent protein, into the muscle of mice, because that's what we thought might be a good route of delivery for the LPL gene in the future. But it really didn't work very well. Essentially, all the green cells were just along the needle track of the injection. And so we went back to the drawing board. I saw a great poster at a conference of people using different serotypes. And so we tried a bunch of different serotypes, and we settled on this serotype called AAV1, which was, if you go back to this previous graph, it was actually by far the worst one in vitro. But in vivo, it was by far the best. And in fact, it was so strong, you didn't even need a fluorescent light. You could literally see green glowing out of the leg muscle. So I settled on AV1, but did learn an important lesson that I think is still applicable, or is really driving my research today, is that the in vitro results were not predictive of in vivo. In fact, they were the opposite. I mean, if they were consistently the opposite, then that would be great too. We could use it, but they're not correlated. So we developed an AAV vector. I won't go into this too much, except that we did some initial studies in mice, trying different doses. This graph here is showing the control mice and the level of their triglycerides, and it's this white line, and it's around 10,000 mg per deciliter. And on this log scale, you can see the light blue is what a heterozygous mouse would have. And so most humans that are heterozygous would never know that they actually carry one of these disease-causing mutations, because they're essentially normal. And the wild type is around 50 mg per deciliter. And so we used a low dose here, and it actually resulted in quite a striking impact, a 77% reduction in the triglycerides, but still definitely quite a bit above the heterozygous range. And in the high dose, so a 10 times jump, the triglycerides now were reduced by 99%. And so that was within the heterozygous range, and that was the ideal goal. And so it works very well. You could literally see it, even by just looking at a mouse. If you turned it over and looked at its leg vein through the skin, you could see the difference between a mouse with high triglycerides and a mouse with low triglycerides. But these are just some photos of the plasma. Made it for a nice picture on the cover when I finally published it. And it was permanent, at least in a mouse. It lasted here. You can see there was no reduction in effect after 1 year. And that's a long time in a mouse. And then we did a proof of principle study, because there was this naturally occurring cat model of this disease, which was quite fortunate, because the regulators really were interested to see that we could do the same sort of treatment in a different species. And so it worked very well in the cat. But one important lesson learned here was the cost of production. And this is also a very important point that I'm going to be coming back to. And at this point, the cost was over $100,000 per animal. Quite stressful for a postdoc to be doing this. But by this time, we'd already, or my supervisor had already helped establish a company to really commercialize this. So that was the only way that we were able to actually move this forward. And so based on those mouse studies and the cat studies, and then some really comprehensive toxicity studies, a phase 1 study was initiated. And this was initiated in the Netherlands, because one of the other postdocs of my supervisor had gone on to become the head of the lipid clinic in Amsterdam. And so we had a lot of patients to work with. And he was the one who really spearheaded forming this new company based in the Netherlands called Amsterdam Molecular Therapeutics to advance this work. And now, so we were producing this virus at the time it was considered fairly state of the art, and what are called roller bottles to minimize the amount of media, but it was just so expensive to produce. It was definitely over $1 million per patient for even just a low dose of this AAV. I didn't go over those results, but that study was good enough to allow us to then proceed to a phase 2/3 study, and this one was based in Canada. But so the phase 1 studies showed that it was safe and effective enough to move ahead, but we wanted to move up to higher doses. And so again, the cost was very high. So even though we were using 10 times higher dose, the company AMT had learned how to make the viral vectors a little bit more cost effectively, but it was still about $600,000 per patient. And so these are showing some results of that on the triglyceride side. So the triglycerides were reduced about 31 to 50% depending on different doses, and overall safe. And so these patients were then followed up for by 2016 they'd been followed up for 6 years, and it, in summary, was that it had prevented all serious pancreatitis. It had prevented or it had significantly reduced the use of healthcare resources. So it was going well. And then in 2012, this drug became approved. It was quite a process. And in fact, the AMT company eventually ran out of funding to try to keep getting, after multiple failed attempts of getting regulatory approval. And it's because it was quite a novel way of treating patients using gene therapy, and this hadn't been approved before. Luckily, another company sort of took up the took up the rights and purchased the rights to this and was able to get it approved in 2012. So it was a great achievement. It's considered the first gene therapy to receive clinical approval. It was definitely a road blockbuster. It had a very difficult time getting through the regulatory framework. But it's also was so rare. It's to treat such a rare disease that the cost that the company had to estimate was they estimated the cost was going to be a lot. So they were charging about $1.6 million Canadian. I mean, that would change based on the exchange rates, but it was a lot of money. Patients, some of the Canadian patients noted, how important this was, how effective it was for them. This patient was able to have 2 babies without any complications, which is rare. And just some of her quotes were just that, she had suffered from this debilitating pancreatitis and had been hospitalized more than 40 times. And then after this, she never had any more pancreatitis. So it was wonderful. It's, really good. I mean, definitely too expensive, at least based on the current sort of system, but no one could afford to pay for this, at least not with a single upfront payment. And so in 2017, 5 years later, the drug was withdrawn because nobody was paying for it. And so the CBC picked this up. They noted, we'd spent a lot of time and money and effort to make this, and it was no longer being there to benefit patients. So the biggest limitation was cost. There was really no mechanisms in place to pay for this. And so, why should we care about cost? There are many now new drugs coming on the market that cost a lot more than this drug. And I put this slide in to sort of give some perspective in that Canada's prescription drug spending in 2020 is about $33 billion. And since this drug was approved back in 2012, there's been 34 new gene therapies approved, and some of these are more than $2 million a patient. And there's now 679 more gene therapies currently in the FDA clinical regulatory pipeline. So these are actually in clinical trials. And there's probably, the FDA estimates there's about 1,500 more gene therapies that are in the sort of preclinical phases that have already started discussions with the FDA. And so there have been some health economists trying to conservatively estimate what gene therapy will cost in the future at this rate. And they've estimated in Canada it might cost $30 billion a year. And so we could either double our healthcare spending. I mean, we already spend a lot of money on healthcare. We could double that spending just to cover these gene therapies. We could try to lower the prices of these drugs, which would be excellent. A third option, which sounds a bit crazy, is that we could literally just hold lotteries for access to these medicines. And ethically, that sounds awful, but this is in fact actually what is happening. In the UK, there's a treatment for SMA, and it's an AAV type gene therapy called Zolgensma. And they offer a lottery each year for 180 free doses of this drug, because normally it would cost $2 million. And so this is basically a worldwide lottery trying to give access to this, it is a life-saving medicine, because otherwise these patients will die. But the ethicists have assured the company that this is a good approach, but I'll leave that for you to decide. So Francis Collins just even this year said we really need transformative approaches so that it's less painful to bring these new ideas of gene therapy to the clinic. And so I wanted to just really briefly touch on two sort of avenues that I'm been looking at. So the first one, I'll just really give a very brief summary, but it's a very exciting approach. And the second one is really the purpose of my presentation today. But so the first one is we're trying to redevelop a less expensive version of that AAV1 for LPL. And this was brought about by that CBC article, and researchers at the NRC were interested in seeing if we could basically reinvigorate that research project. And so it is underway, and it's going very, very well. And I think what's even more important than that single new version of AAV for LPL is that it has a much bigger goal, and its goal is to really try to develop new models where we could develop and pay for innovative medicines like this gene therapy and others in Canada, and how we could maybe manufacture these in Canada and offer them to patients basically at cost, which would be just fantastic. And this is being led by Dr. Danica Stanimirovic from NRC. And so how can we actually get these AAV gene therapies to be lower cost? And so some of the ideas are that we could try to increase the scale of manufacturing. And we think by doing that, we could lower the cost by about tenfold. Public supported manufacturing and even centralized facilities that many different companies or universities could access to develop to produce these vectors for clinical use. And then the last one is been what I've been working on is basically updating that formulation of Glybera with 20 years of new technology, and we're making it a much more efficient AAV. So the other approach, this is why I'm here today. This is trying to really a whole new version of gene therapy, and this is for gene editing and using lipid nanoparticles, especially because they're so ideally suited to this. And so I'm going to tell you about a project that's underway that I'm working on with Peter Cullis and the NMIN nanocore team, and Dom and Jay from Nanovation, and Star Lee and Sarah Hedrick from UBC, as well as with David Liu from MIT. And our goal here is really, can we optimize in vivo therapeutic genome editing using lipid nanoparticles? And so before I go any further though, you might be wondering, like, why genome editing? And believe me, I've thought a lot about this as well. And because that standard gene augmentation, I mean, I showed you just some really, really striking results. And I think that approach could be applied to literally, more than 1,000 different diseases for sure, and have huge benefits to patients. So some of the pros and cons is that, I mean, a real pro is that it's transient for some of the lipid nanoparticles to deliver mRNA. And that can be a very useful capability, or it might be permanent if you use something like AAV, and that has good and bad things about it. LNPs are really well designed, or at least they have the ability to redose with them, and people have shown that with Onpattro, the first LNP to be clinically approved. But some of the cons is that it really doesn't restore the precise gene regulation. So this is really important for many different diseases, essentially all dominant diseases, and many other recessive diseases as well, where the mutant version of the gene might be causing more problems than you would want. Other problems with AAV is that in some cases, the integration of the genome has been shown to cause some genotoxicity, which could lead to cancer. And we've seen that now in newborn mice that are treated with some AAVs. They cause hepatocellular carcinoma. So that's a very concerning risk. AAV are also limited to just a single administration. Once you use one serotype, you could never re-administer with that same serotype, or at least not with good efficacy, or not as good. And then the AAV has quite a small size limitation, and there are some tricks to working around that, but every time you try to make it a bit more complex, the efficacy will be reduced. So why genome editing? So it is permanent. It's essentially just you take away all the complexity of gene augmentation, and you just get right to the root cause of the disease, and you're trying to fix that underlying mutation. So it's permanent. It would restore the precise regulation of the disease-causing gene, and what I'm excited about it too is that once you can do that really, really well for one disease, you should be able to use almost the exact same components to treat almost any disease-causing mutation out there, and that could even be applied to cancer, trying to treat early cancer mutations. If you could do it really, really well. But the con is a big con, and it's the potential off-target genome edits, which would cause genotoxicity, which could be very bad. So the big picture, they both have pros and cons. They both have different applications, but I'm excited to talk about genome editing because I think it has some advantages that really can't be overlooked. So a little background on genome editing. So CRISPR really was a massive leap in our ability to do genome editing, and the 2020 Nobel Prize was awarded to Emmanuelle Charpentier and Jennifer Doudna for their discovery of applying CRISPR to genome editing, and they showed this in E. coli cells, and then a year later, Feng Zhang and his colleagues at MIT were able to do this in human cells and in mouse cells, and so that's sort of the background of sort of standard CRISPR. So how does that work? I know that most of you are probably familiar with this now, but just to sort of set a level playing field, you essentially have two key components, this CRISPR-associated protein, and most commonly people using this Cas9, and then a guide RNA. And these used to be multiple different parts, but people have simplified it to a single guide RNA. And this guide RNA targets the Cas protein to a specific region on the DNA based on the guide RNA sequence. It binds, and the CRISPR-associated protein cuts the DNA. And that's standard CRISPR. And then what do we do with that double-stranded break? Well, one thing is you could just do nothing. You could just leave it and hope that the cell will not be able to repair it quite as precisely as it might want. And so you end up with deletions, or you end up with insertions. So that's a great way to introduce random errors and maybe knock out a gene. It's not precise. That's the downside. Another option that you can use that double-stranded break for is you can do what's called homology-directed repair. You basically flood that cell with a lot of new DNA called donor DNA, and the cell will try to repair that cut, and it may use that donor DNA as a template. And so if that happens, you can kind of trick the cell by putting something else in that donor DNA, and the cell will incorporate that change, and now you've modified the cell. And what you've ended up with by doing is a precise edit, but it's not that efficient. And so the people are beginning to get better at it, but in general, the efficiency is typically less than 1%. So the big limitations of this is that it causes these double-stranded breaks in the DNA, and for me, that's really essentially a non-starter. Anything that causes double-stranded breaks in the DNA is not going to be safe enough for real, I think, for clinical use. And then the low efficiency of HDR is yeah, we're going to want something that's works a bit better than that. And then, of course, the problem that will apply to all genome editing is the problem of off-target genome edits. But there's been a lot of advances in this field, and I'll just briefly touch on a few. So there's so many different types of novel Cas proteins from other species, and these are allowing researchers to be able to target more and more regions in the genome. And people are modifying these Cas proteins, making them more specific or less specific for better or more broad genome editing. And what I wanted to really focus on is what I've been working on is these quite I guess like kind of a second generation of CRISPR genome editing, and it's using these things called base editors and prime editors. And to keep it simple, I was just going to describe how a base editor works. And these are, so this is showing this Cas protein, but it's got a D in front of it to signify that it's been deactivated. And so it's this inactivated Cas9, and it's linked at the protein level or at the DNA level, if you're making it from mRNA or cDNA, to a DNA modifying enzyme. And this one is a deaminase. And so how does this base editing work? So again, you only have the two components. You have this sort of fancier version of the Cas protein with this DNA modifying enzyme, the base editor, and again, a guide RNA. This guide RNA targets the base editor to a specific site in the genome, and it brings this DNA modifying enzyme into contact with the DNA, and it causes a precise edit. You can calculate, where it will occur exactly in relation to this target sequence. And so working backwards, you can design base editors to precisely fix an exact mutation. And how it actually modifies the DNA, this is an example of a cytidine deaminase, where it deaminates this cytosine and turns it into a uracil. And at the same time, it's a little bit tricky, but it nicks the other strand, the opposite strand of DNA. And so the cell thinks that the cell notices these don't match. And it thinks that an error has occurred on this strand of DNA. So it preferentially repairs that strand. And so here it's pairing up an adenine with the uracil, and then this thymine gets incorporated eventually. But it might sound a bit complex, but it's just a beautiful system. And the big thing about it is that it's just so much more efficient than the sort of standard homology directed repair. So people typically can get 50% gene editing in cells, and in some cases people can get much higher levels of gene editing. So going to our research question, so can we improve the effectiveness and safety of genome editors for in vivo therapeutic genome editing? And for me, it's where to start. There's a million things that we could do, and I'm always torn between safety and effectiveness. And there are so many things we can do. We can try to maximize the expression of these genome editors to improve efficacy, but that would come at a cost of safety. Maybe we might be more interested in like what specific cells we could get them into. Anyway, there's a lot of things to do. And so on the safety side, my lab has been working on developing models to really cost effectively be able to measure off-target edits from these new types of genome editors, because there's a lot of off-target a lot of ways to measure off-target edits using more traditional standard CRISPR, but not for the base editors and prime editors. And so we've been able to make these model systems to do that. But I won't talk about that today. But on the effectiveness side, what we've been sort of playing around with is trying new formulations of base editors and prime editors in collaboration with groups from the US. And it's going very well. I mean, that you can certainly improve the efficacy a lot, but we're worried about the improvement at a cost of safety. And how we're delivering them right now, at least we're really focused on mRNA. It's a really good strategy to deliver these different formulations into cells. But what I'm personally really most excited about is the work on the delivery side. And so I've crossed out here AAV because, just as a reminder to say how, the long-term expression of a gene editor from something like AAV is just not appropriate. And like I said, AAV is really appropriate for certain types of gene augmentation. But in this case, it's not, unless it's somehow regulated and you can turn it off completely, because you don't want that genome editor in there continually trying to edit. You want it in there literally just for maybe an hour or two, however long it needs, just to do its edits, fix that cell, and be done. So we've been working with lipid nanoparticles to deliver these different cargos. And so as I mentioned, these are base editors and prime editors. Really briefly, what the difference between a base editor and a prime editor is that the prime editors are a little bit more complicated in that they use a reverse transcriptase, and they basically write in a new piece of DNA at that exact target sequence. And so essentially you can introduce any new nucleic acid sequence at that site, and they don't have to be even a single nucleotide. They can be 40 base pairs, 80 base pairs, and some new research is showing huge sizes, up to thousands of base pairs. And anyway, so that's prime editing, but essentially this doesn't work nearly as efficiently yet as base editors. And so this is just to remind me to tell you about, when you read about other base editing things coming out, there's so many different types of base editors. This is just a snapshot from back in May. And so the number of targetable sites that you can target with a base editor is increasing a lot. But there is a downside to base editors in that each is designed, you would set one up to target a specific single mutation. And so that's the big advantage of prime editing, is that you could instruct it to essentially rewrite an entire exon of a patient's genome as a way to maybe treat many different mutations causing a single disorder. And these example papers are just showing that there's a lot of work going into improving the efficacy and safety of prime editing, and it's gotten a lot more efficient than when it first came out. And so a lot of groups are actually seeing like are able to get levels of prime editing, more than 50%, even more than 90% of the targeted cells, which is wonderful. Okay, so back to our research question. Oh, delivery. So remember, my big lesson learned was that the in vitro results don't necessarily correlate with in vivo. And I've worked with some wonderful people here at UBC, helping us really take the next step. And I saw Dr. Dahlman's talk just a few weeks ago, and he was showing us again how in vitro LNP delivery really is just like these viruses, was not predictive at all of in vivo delivery. And so this is just one of his scatter plots that he showed for many different tissues. And so again, we really have to keep in mind that the lipid nanoparticles won't be predictive in vitro compared to in vivo. So we thought of a way of how we could do this, and what we wanted to do was come up with a mouse model that would allow us to test different lipid nanoparticles for in vivo readout. And as a PhD student, when I just remember how difficult it is to actually quantify something in vivo and to see where it is in different tissues. And so I really wanted something that was simple, and you could almost, wouldn't it be great if you could just see the whole mouse and see where something was, and wouldn't it be wonderful if it was even from a living animal, then you can measure them in multiple times. And so that's what we've created here. So we started with these wild type mice. We're able to get a luciferase positive constitutive mouse by modifying a mouse from Jackson Labs. And we introduced a mutation into these mice from Jackson Labs. And this mouse is one that would be targetable by the adenine base editor or prime editors. And so this is what we call our luciferase reporter mouse. So the goal is to basically be able to have this mouse and see where, if we can ever fix that mutation, it would turn on luciferase, and we would be able to then image it in a system like an IVIS animal imaging system and see exactly where we've fixed that mutation. And so the goal was that we could make something that we could quickly test different lipid nanoparticles and try to optimize things to work better in vivo. And of course, a mouse is not going to be completely predictive of a human or even of a larger animal, but it's to me infinitely better than doing it in cells. But of course, we have to make sure that these lipid nanoparticles first work in cells, at least at some basal level. So anyway, so where are we now? So we've been able to make these luciferase constitutive mice and then introduce a mutation that knocks out that signal. And so this is our best mouse so far, an ADE mouse that has almost no background. And so the next question then was, well, can we actually fix that mutation? And so our sort of proof of principle studies was using those clinically approved lipid nanoparticle formulations like Onpattro, and see if we could deliver the mRNA for the base editor together with the guide RNA. And this was then, of course, targeted to that exact mutation in the luciferase gene. And so we were able to do that. I was actually quite surprised how well it worked, because we were able to get over 90% of the cells in the liver, at least based on the signal, to be restored in producing luciferase. And it's a very striking signal with a I guess a moderately high dose, take 2 mg per kg, but it's not super high. And the neat thing is, if we ever wanted to increase this, we could add more, we could re-administer, but if we're already at 90% for the liver. But this result also really highlights the problem that we're now really trying to overcome, and that's that it's only in the liver. And of course, that's what these lipid nanoparticles were designed for. And at the recent Liposome Research Days, Peter Cullis gave a wonderful talk about sort of the history of these nanoparticles, and just and so many people ask him the question of why are these only go to the liver, and he was just gave a great sort of background about it, and really the answer is that they were designed to go to the liver. And so our question is, can we modify this great delivery system, and can we get them to go to other tissues? And so my focus has been, can we get these at least to get into the muscle. I mean, the muscle makes up 40% of our body mass. Wouldn't that be great if we could get these into there? Because also, you know, from a disease perspective, there's a lot of muscle diseases, and there's and lipoprotein lipase deficiency. We know we can treat that by treating the muscle. And so we, again, did a very similar study using these mice, but now a much lower dose, but only injecting them into the gastrocnemius muscle. So basically your big calf muscle in a mouse leg. And you can see that, you know, within 2 days, you have a very strong signal, and it's there after a week, and it's there after 4 weeks, and it's there after 30 weeks. And I recently gave another talk about this, and one of the audience members was confused as to like how is that expression lasting for so long? And it's just important to reemphasize that, you know, this luciferase gene in these mice, it's in every single cell, but it normally has that mutation. And so we've just been able to, what this shows is that we've been able to precisely fix that mutation in just in those cells to turn back on this signal. So there's no it's very different from a simple mRNA delivery. You've actually precisely fixed that underlying mutation. So it's not all great though. On the downside is that we're only at about 2 to 3% editing in those muscles. So these pictures look beautiful, but we have a long way to go. So, but it's still actually far better than I would have imagined based on, you know, lipid nanoparticles designed to go into the liver. And we're working with Peter Cullis's NanoCore team and Nanovation to develop sort of new formulations that may be better to target muscle. Okay, so I'm close to the end of my presentation. So this is, to me, the bigger question. You know, what cells do we really want to get into, and how many of them, and how many do we need to correct to treat a genetic disease? And so the human body has about 37 trillion cells. You know, for LPL deficiency, based on our work with AAV, we know that, you know, where we're at already with muscle, if we can repair 3% of the cells, this would have, you know, huge clinical benefit to patients. It would probably reduce their levels of plasma triglycerides, you know, far below any sort of toxic levels. But again, I think we can do a lot better than 3% if we can, you know, even slight improvements on these lipid nanoparticles will really have huge improvements. But yeah, every different disease has different needs, and truly one of my goals is, you know, why can't we just try to, instead of trying to make nanoparticles that target this tissue or that tissue, which of course has wonderful uses, but I'm really interested in just trying to get nanoparticles that will go to almost, well, hopefully every single cell in the body. That's the geneticist's dream. If we can fix those mutations in every single cell, you'd basically turn a patient that has this disease-causing mutation in every cell into a patient that doesn't, and it should, you know, be able to fix essentially any genetic disease. And again, going back to sort of the rationale is that, you know, all of these completely different diseases, they all again have the same underlying cause of mutations in different genes, and all of these people shown here are really pioneers in gene therapy. So I think Cynthia was one of the first Canadians to receive the AAV gene therapy for lipoprotein lipase deficiency. Michael, I've worked with his parents, and they've raised millions of dollars to develop an AAV-based gene therapy for his disease, and now he's been treated for that. And Makeda and her mom are trying to raise, again, millions of dollars to treat her disease using an AAV-type approach. And Jeff has dedicated his entire life to trying to treat Huntington's disease. So I really think that there's a lot of potential for genome editing to treat all those different genetic diseases. And in the future, if we can get it to work well enough, we could potentially treat cancer, you know, even before it's really causing too much problems. I mean, it would have to be hit pretty early, and maybe even infectious diseases. So just to summarize, I'm really excited about genome editing because I think it has the potential to treat thousands of different diseases, and yeah, I would love to see in the future being able to just target to get these into every single cell in the body. So that's my presentation, and I would just like to acknowledge, you know, the wonderful support and work from everybody in my lab, and especially from our collaborators. and I'm really excited about our new NMIN funding project on the gene therapy grand challenge team. And this is a wonderful team with Anna Blakeney and Peter Cullis and Sabrina Leslie and Ken Harder. Those are the sort of PIs, with a wonderful team across Canada and in the US, to really take this to the next level. We're really excited about it. But I'm happy to take questions. And
thanks very much, Dr. Ross. That's been an amazing talk. The kind of talk that I think one sees once every 6 months or something like that if you attend conferences regularly. So I would like to open the floor to questions from participants. You're all presently on mute, so to ask a question or to join the discussion, please use your control panel to either raise your hand to speak or submit a written question. I'm going to pause now to see if Marshall has something in his hands.
Yeah, we have one participant with a raised hand who is Syed Abbas. So Syed, if you unmute yourself, you should be able to talk now. Please go ahead.
Hello.
hi, Syed.
I am Syed Abbas, a postdoc at the Johns Hopkins University School of Medicine. Yeah, impressive work. I'm very interested about the reporter mouse model. I'm just curious to know why did you choose it to be luciferase instead of a fluorescence intensity kind of signal?
Yeah,
and
no, yeah. Oh yeah, I won't cut you off. I'm sorry. I was just teaching my PhD students this morning. Don't cut off the questioner before they finish. So you finish your question, please. Sorry.
I know that was the question, because I mean I just would like to hear from you what
Yeah. Oh, happy
to.
You know, honestly, we made several different models, and some of them were with GFP, some of them, the other ones were with luciferase. I don't know which to hit first, but I really like luciferase because I can see you know, essentially every tissue in the body with that IVUS imager. So to me, that's a real bonus. But at the same time, it's not, at least not today. I'm hoping within a few weeks we'll be able to, you know, really narrow in on these tissues more precisely using, you know, immunohistochemistry. But you know, a big advantage of something like GFP is that you wouldn't need that immunohistochemistry. You could just take out the liver and freeze it and section it and look under the microscope. That would be perfect. I think one of the issues that we came up with on the GFP side that kind of halted that side of the project was that the mice that we received from Jackson Lab that had the GFP in all cells had multiple copies of that gene. So we were able to successfully introduce mutations into it as we wanted, but they still glowed green because they still had other copies of the gene. So that was a bit of a disappointment. If I was gonna do that again, I would have just probably done what I did with the luciferase, is basically introducing it in the Rosa locus. You know, just take a wild type mouse, introduce the GFP gene with your mutation, you know, one with and one without, and that probably would work better. So I think that would be a good approach. Except, yeah, I think just the luciferase, I love it, and I'm such a fan that I've convinced my faculty to buy a new imager, which I'm just so thrilled about. So we're going to be able to even detect this luciferase even with better sensitivity. But those same imagers can image fluorescence. So you know, I think it's, yeah. I ended up going with luciferase because the GFP stuff didn't work. But yeah, does that answer your question?
Yeah, great. Thank you very much.
Yeah.
Thank you, Syed. Great question. Marshall?
We have a written question that I'll
read.
read.
I see that. I can read it out.
Go ahead.
From Xin Yi, it's expected that the LNP with IM injection, or is it expected that the LNP with IM injection would show no liver accumulation? Now, I really love this question because we've been following up on this as well, because I was interested in this. And so in follow-up studies that we've done with higher doses, we are seeing liver accumulation. So I think it's a great question, and we are seeing it leak out of the liver when we use higher doses, and it does then turn up in the liver. Yeah, you're welcome.
Thanks a lot. Marshall?
There are no other raised hands or written questions at the moment.
So may I ask a question, Dr. Ross? May I
Yes.
ask, what do you think are the prospects for the single base editing compared with multiple base editing? Like,
Yeah.
how long do you think it will take for the first to make it to the clinic? And should we like really focus our efforts there? What do you think?
Well, I think on the base editing side, it's already now going into the clinic, which is exciting. They are using it to target that PCSK9 gene, you know,
Right.
sort of a cholesterol-related one. And I haven't seen the clinical results yet, but I've seen the some of the large animal model results, and it's really striking. And my lab is a little bit jealous because they're saying how, well, those guys have it easy. They just have to introduce a stop mutation anywhere in the gene, and they can get their effect. Whereas I, you know, give my students a high bar to reach, which is, you know, here's a mutation that you need to target. We need to fix it. You know, fix it. And that's tough. That can be that doesn't always work so well, at least with today's current base editors. But so I'm excited that base editors are now entering the clinic, at least in clinical trials. But on the big question of the sort of prime editing versus base editing, I'm really excited about prime editing. You know, I haven't been able to achieve the high levels of prime editing that some groups have seen, but that's because, you know, some of the more recent papers that have come out, they're using just amazing tricks and to maximize the levels of prime editing. And it's hard, honestly, to keep up with it because the people are introducing so many new things, and my poor new grad student has already been working with me for more than a year as an undergrad, trying to keep up, and every time he catches up, a new thing has come out. And so I think, like, you know, honestly, as long as the safety side is going to be the same for prime editing as base editing, then I think it could probably take over from the base editing. But yeah, today base editing is still considered quite a lot more efficient. So, but yeah, just the potential for prime editing is massive.
Yeah, it's gigantic. Thanks for that answer. I have a personal curiosity about the CFTR gene, so cystic fibrosis gene, which is potentially
yeah,
that's where does
such a
that
good one.
stand? Where does
I
that
don't know
stand?
where that is but you know, is that a three? Is it a single nucleotide deletion that causes that, or I forget?
Yeah.
Is it? But that would be a great. I don't know where we're at, where people are at on that, but
I see. But it seems like it's definitely a valuable target. There's no doubt.
yeah.
And it's also varies. There's patient stratification that's necessary, of course, as well.
Yeah.
I see a few questions in the Q&A.
Yeah, I can read these out. So, interesting talk. I have a question. I'd like to use the lipids of Onpattro, like the ones that we use as a control, kind of gold standard, to compare them with new nanoparticles that we're developing. Are these lipids easily available? Should I contact the company? So this is a great question, and you know, today this talk is sponsored and being held by NMIN, and what I think was just one of the most amazing and brilliant things was that I remember in some of the early planning meetings about NMIN, and they said, well, you know what, we one big problem out there is that people report on using the same type of lipid nanoparticle, but it's actually made quite differently. And so NMIN created this group called the Nano Core, which is led by Peter Cullis. And for me, I just go to the Nano Core and I say, you know what, guys, I need some of this Onpattro type formulation, and they make it, you know, to such professional standards. I mean, Peter Cullis is really the developer of lipid nanoparticles, and so to be able to get them from him and his lab is wonderful. So if you're not part of NMIN, you could probably I think that they have avenues for non-NMIN researchers to reach out to them and work with them for sure. And if you already are part of NMIN, then you're already all you have to do is email them, and they're the experts on where you should get it from. Okay, so the next question is, how long does the mRNA stay in the cells, and are there immunologic concerns on the base editor proteins? So this is a great question, and one that I honestly don't quite know the answer to. How long does the mRNA stay in the cells? I was just at a defense this morning, and we were commenting on how long like siRNA stays in the cells, and or at least has its effect, and it's, you know, even on patro, I mean, it basically, you know, it lasts for weeks. I don't know how long the mRNA stays, but that's a great question. The way I think of it right now is that it probably stays in there for, you know, 24 to 72 hours, I think, is what we have seen with looking at delivery of things like luciferase and things like that. But, you know, probably it stays in a bit longer than that. And are there immunologic concerns to the base editor proteins? That's a great question. And there was a recent sort of, I think, Nature Communications paper where they injected mice with not a base editor, but a different type of sort of CRISPR, more traditional CRISPR, and they were able to show that that they could repeat administer it. So this is an important question. You know, can we dose multiple times? And I'm hopefully, I mean, I think these mice that we have would be a great way for us to easily answer that question. So but at the moment, from a single administration, I mean, I was quite concerned when we first injected these mice that maybe after, you know, 2 to 3 or 4 weeks, we might see a drop in signal if these mice developed an immune response against this new, you know, fixed luciferase, but they don't. So that was really lucky, I guess. And do you have any thoughts? So this last question is, do you have any thoughts how results achieved with this rodent model may translate into large animal models? Well, I'm optimistic that they will translate well, but we'll just have to see. I was on a PhD defense with one of Christian Kastrup's students this morning. It was really well done defense, and they were comparing results that they had achieved in mice and then in a large animal model, and it translated very well. Okay, one more question from Kevin Fox, someone whose work spans the field from viral to lipid nanoparticle delivery. I'm curious on your thoughts about the hybrid approach featured in the 2019 article by Sangamo and Acuitas using zinc finger nucleases delivered by AAV and template by LNP. Yeah, I remember seeing that. You know, if anyone, there's no one in the world that I think that loves AAV more than I do. It's a really amazing virus. I'm not such a big fan of it for genome editing because I worry about it being in those cells for such a long time. And I guess it depends on what you're delivering by that AAV. But you know what, maybe another thought is that, you know, it's a really good lab tool to show proof of concept. And so, you know, because it's so good at delivering things into cells. And so, yeah, I think I don't think I would want to, if it was me, I wouldn't want it to be delivering a genome editor into me. And the anonymous attendee about other nanoparticles. We've been working with some amazing collaborators here at UBC. You know, I'll just list Anna Blakeney is a wonderful, relatively new researcher at UBC, and she has an engineering background and has been introducing us to a lot of different new types of nanoparticles, and it's been sort of fun. But we don't have anything to present yet. But some work, some don't. So that's all I can say.
Yeah, I think at the moment the real strength is at least in Western Canada is with these LNP systems, and there's so much opportunity. People are really trying to figure out what to do with them. There's just open vistas.
It really is. I'm really I think I emphasize it, but what we can already achieve without, I mean, I say without trying, but really, like, you know, Peter Kellis worked for 40 years and is still working harder than almost anybody I know to make these nanoparticles better. But he really made it easy for us to be able to achieve such high gene editing in livers in these mice. So there is so much. I mean, there are thousands of diseases just waiting to be treated from that type of approach. That it's it's a bit crazy that I'm even looking at other tissues, you know, muscle and things. I mean, we should really just be focusing on liver. It's such a low-hanging fruit, and there's so much to do. So I really encourage people to do that. I mean, it works very well.
Works very well. Liver is a bioreactor. Just send down
Yes,
that.
exactly.
Yeah.
Peter wrote a beautiful review article a couple of years ago about that. If anyone's interested, I can send them that version.